Good afternoon, everyone. Welcome to the H.J. Barnett seminar series that we run on a monthly basis. And today is February, so this is the second webinar for the year. And we are honored to have Professor Ozlem Bishop to be the webinar presenter for today's webinar. And I just want to actually just say a huge congratulations to her because she recently got promoted to full professor at Rhodes University, and I think that's a mean feat. That's a huge achievement, and I just want to say, well, obviously, besides the fact that this is a webinar and it's about her work and her passion, the reason why obviously she got promoted is because she's been doing such amazing work, and I just want to say congratulations to her. And you can join me later to congratulate her as well. But just to give you a bit of background, and I'm not going to even say the whole biography because it's a long list of achievements. It's an amazing CV, but I'm not going to read the whole thing. Just know this: Ozlem is well educated and well traveled, and she's passionate about bioinformatics in Africa. Her broad interests, or rather research interests, include structural bioinformatics and bioinformatics education. She's a member of multiple international educational bodies in bioinformatics, such as Goblet and ICB. She's also an honorary member of Golden Key, and she's a co-founder and the president of SASB as well. So I think if you want to read more about her, I think in the link when I was advertising her webinar, I did put in links of the institutions that she works at, such as Ruby and Rhodes as well. So if you want to read more about her and her work and her publications, you can go to those websites. But for today, as you can see on the first slide or the main slide, she'll be presenting a talk titled or webinar titled Integrated in Silico Approaches towards drug discovery, some applications to allosteric drugs, pathogen resistance, and precision medicine. You all had access to the abstract, but I'll just read literally just a bit of it, just so that we know what the talk is going to be about. The presentation will summarize the workflow that Ozlem and her group and related tools that they develop mostly under HUMA, which is a Human Mutation Analysis Portal, the project that Ozlem and her group have been working on for a couple of years now. Grand Challenges Africa, the drug discovery program, as well as various National Research Foundation of South Africa NRF grants. The presentation will include multiple examples that Ozlem and her group work on. So without further ado, I don't want to waste time trying to introduce, obviously, a woman of her caliber. But I just want to say thank you so much, Ozlem, or rather Professor Ozlem, for agreeing to give this webinar in February. The mic is yours.
Okay, thank you, Pablo, for a very nice introduction. And before I start, I would like to thank H3 Abionet for giving me this opportunity to present our work here to you. Today I would like to talk about some of the approaches and tools that we have been developing in the group with some applications to drug discovery related topics. But before starting, if you don't know us, I would like to give brief information. We are at Rhodes University, which is located in Grahamstown. It's a small university town in South Africa. The main strength of Ruby is the postgraduate studies. We have currently roughly 30 members. Most of them are PhD and MSc students. Our main interest is in structural bioinformatics. Structural bioinformatics focuses on the identification and analysis of structure, dynamics, and interaction of biological macromolecules in 3D space. And while doing this, it aims to make connection between sequence, structure, and function. And the structure is particularly important because if we have structural information of a macromolecule, actually we can derive a lot of information about the function. But most importantly, 3D structures are crucial in structure-based drug discovery process. We have a number of examples from FDA-approved drugs that they are actually designed using the 3D structures of protein targets. Today I would like to divide my talk into two parts. The first part is mutation analysis. A bulk of the talk will be on that. And in the second part, I briefly will talk about the allosteric. And the research questions that we will be asking in the first part is what are the effects of missense mutations on protein structure and how can we analyze them? These questions are actually highly important to understand the underlying causes of many inherited diseases, to investigate drug sensitivity issues in certain populations, hence for the precision medicine purposes, and to uncover drug resistance mechanisms. For the allosteric part, the main question that we will be asking, how can we identify alternative drug targeting sites? Okay, let's start with the mutation analysis. Our main role in H3 Abionet, as Paolo mentioned, is to establish tools to do downstream mutation analysis. Hence, we first established HUMA, and HUMA is a platform for the analysis of genetic variations in human, and it's a database and web server. HUMA basically integrates sequence structure variation and disease data into a single connected database. It is very user-friendly. You can do many things by click-based form. HUMA also has some tools, and we're currently developing more tools to analyze the variation data. Furthermore, I think this is very important for H3 Africa users. Users can upload their private variation data sets securely without sharing with anybody, which are automatically mapped to public data and can be analyzed using the integrated tools. One of the tools that is integrated to HUMA is Primo. In this project, besides David is the, okay, let me show here. David is the main, it was the main person establishing HUMA. Primo was established by Rowan, David, and Michael. And basically, Primo is an interactive homology modeling pipeline. So people can actually use Primo, which is linked to HUMA, to model their proteins with the interest of mutations. The other tool that was developed for HUMA was Vapor. We have a number of web servers looking at the SNP effects, and Vapor basically aims to bring them together under one format. So you put your variation data in one format, and in the background it goes and calculates for each of these tools and returns back to you a consensus results. The results are presented in this form. Perhaps it's not clear here, but the first part is predicted functional impact, and the second part comes as predicted change in stability. Okay, so, but we wanted to actually incorporate more tools to HUMA, especially to analyze the missense mutations. But when we started looking at this problem, we realized that actually there is not one well-established protocol or one established tool to do that. So we decided to propose our own protocol, and this protocol actually published in 2017 in a review article. This was the H3 Africa special issue in Global Heart. So what we did basically in this protocol, gathered a number of available tools, but more than that, and these tools were basically, the analysis was basically finishing molecular dynamics level, and on top of that we proposed something called dynamic residue network analysis. Up to that, actually people were using residue network analysis, but they were mainly using either minimized structures or the crystal structures. What we propose to do here to use the molecular dynamics trajectory data and get a better and more accurate results out of it. So by using this idea, we developed a tool, software suite basically called MD-Task. MD-Task has a number of functions, but in the dynamic residue network section, we provide calculations for various matrices, including the shortest average path and betweenness centrality. I'm just not gonna, there is no time, I'm not gonna enter to the details of these matrices, but basically what we saw over the years, the most effective matrix is betweenness centrality, and it is a measure of how important a residue is in the communication within a protein. So you can think that protein is like lots of residues talking to each other, and some residues are more popular than the others, and they are more talkative than the others, and they are the ones actually functionally the most important ones. And BC basically measures these residues, identifies these residues. Okay, so as a case study, when we proposed this protocol, we decided to look at quickly a system, and that was the renin-angiotensin system, RAS system. This is a very important system, and dysfunction of this system can cause various diseases, including hypertension, kidney disease, and heart failure. What we saw here, it was like a gold mine. There was over 500 SNPs variations actually in this renin-angiotensin system. There was no way that we could look at all of them. We decided to concentrate the interface variations. We selected 15 variations, 6 from renin and 9 from angiotensin, and modeled the proteins. We had 25 mutant models. Some of them included the pairs of the mutations as well. So as a next step, what we did, we looked at vapor for prediction how damaging these things, and according to vapor, most of these variations were damaging. Today I'm just gonna give very quick example to one of them. That's the P40L mutation. So what we do normally after modeling the proteins with mutations, we take them to molecular dynamics simulations to see how the protein behavior is in the cell and how it changes relative to the wild type. As you can see here, wild type and the mutant proteins were behaving differently. And when we looked at the trajectory in molecular dynamic simulations, we saw that actually in the mutant one, I'm trying to get these two proteins, and renin and angiotensinogenin, were separating from each other, and that was basically due to the loss of interaction between these three residues. Right, and we also developed a tool under MD-Task to check the residue contact maps, and this basically looks at the frequency of the interaction. So as you can see in the wild type, the residue proline is interacting with these two key residues 100%, and in the mutant that interaction is mainly lost. So that also explains why these two proteins are separating from each other. So, and we see between the centrality, this is what you are seeing here, actually between the centrality difference between wild type and mutant proteins. And as you can see that these in heat map, and these regions are showing the differences between wild type and the mutant systems. And when we met these regions, and they were basically in the interface area. So what we can say from here, one residue mutation can affect the whole interface of the protein system. And this is from the other protein side. We see the same thing. So the whole communication system in the network is changing. Okay, my next example is from carbonic anhydrases. This is a family of proteins, and specifically here we looked at carbonic anhydrase 2. This work was done by my PhD student, Ellen. The deficiencies in this protein cause osteopetrosis. Osteopetrosis is opposite of osteoporosis. We looked at 6 validated variations here, and the aim here is to understand how these variations are causing to disease, so that we can perhaps then design activator compounds instead of, normally you look at inhibiting compounds to maybe cure the disease. But here, if you understand the mechanism, we think that we can perhaps use that information to design activator compounds to reverse the function of the protein. Okay, we again use more or less the same approach here. Basically, the SNP identification analysis, we again used HUMA and vapor to get the first impression, and then modeling and molecular dynamics combined with dynamic residue network analysis. And this is the protein. As you can see, these actually mutations are, except this one here, are far from the protein, and we suspected that this is allosteric effect. Okay, so when we went to literature and identified all the important residues, functional residues of this protein, and these mutations were not included to this list, then we looked at the short motifs. Short motifs can be functional motifs. And what we saw here, some of the variations are actually included in these motifs. The red ones are the variations, some of them, and they were very close to the functional residues in most cases. And the functional residues are the bold underlined ones. And some of the variations were actually between the mutations. So what we did, we again used molecular dynamics calculations and globally looked at if there is any change. What you are seeing in this figure is the root mean square deviation of the structure from the initial structure. Green one is the wild type, and gray one is the mutant proteins behavior. For instance, this is in totally different space. It seems the mutation didn't globally affect the protein. And we looked at in different states of this protein, because this protein has carbon dioxide and bicarbonate, so we also did the molecular dynamics in different states of the protein in wild type and the mutant form. Okay, so then this is between the centrality calculations. What you see here briefly, the delta BC variation minus wild type, and what we see that actually there is quite a bit of change in the communication. Delta BC increase means that with the introduction of variation to the protein, with the introduction of mutation, actually communication system is changing, and you can see that number of actually the functional proteins are in the communication hub here. Okay, so what we basically were seeing here, that I'm not actually showing the details, but it's all in the article, allosteric effect of this distant variations. This is the variations in distantly related are communicating via important functional residues and changing the function of the protein. So these regions are mainly the active site regions, but also there's a region called primary and secondary aromatic cluster residues, and we see the changes, communication changes in those regions as well. So we think that by using this information from here, one can start actually thinking to identify the compounds binding in this region and reversing the communication. Okay, the next study that briefly I would like to talk is from HIV protease, and this was done by Olivier during his PhD. Basically here we tried to characterize the drug resistance. As we all know, HIV protease is well-established drug target. There are a number of drugs. We looked at 8 drugs here listed with the name abbreviations, ATB, DRV, etc. But each of these drugs, patients, the virus gains resistance after some time. And what you are seeing in this figure is actually these red dots are the mutations due to usage of this drug. And it is not, this is the active site of the protein, and the mutations are not in only active site, it's all around the protein. It almost is random. What we wanted to know here is there a mechanism behind it? It cannot be random mutations. There must be something consistent in the behavior of the protein to gain resistance against the drugs. So what we did for each case, we got 100 highly resistant sequences per antiretroviral, and 100 hyper-susceptible sequence per the same antiretroviral. We modeled them, and then we put them into short MD simulations. Basically, we had 200 times 8 for 8 drugs, and we did this double, repeated twice times 2. We had 3,200 MD simulations, and each MD simulation was 2 nanoseconds, and we at the end had 6,400 nanosecond runs, which was roughly 7 million CPU time. And we combined this data with network analysis and statistical tests. But we did something else here. We didn't use dynamic residue networks. We looked at the residue-residue interactions and looked at the distances, how the change is. And we systematically saw that actually there is a lateral expansion in the protein regardless of the drug. So the expansion is almost in left side. You can see this, and then there's a contraction from the back. It's almost like trying to expand from both sides and pushing from the bottom to dislocate the drug. So we think that whatever you design for the HIV protease, for the active side, virus will find a way to get rid of the drugs. And the better way of designing is to use these regions, allosteric regions, communication regions, to design new inhibitors. Okay, similar study is we currently are looking at in TB against the drug pyrazinamide, and this is a group of students are working here, Rita, Thomas, Olivier, and Sopakama. And just one example, this is unpublished data, but when we do statistically guided network analysis, we are seeing similar thing. There is an expansion in one direction and contraction in the other direction in the protein to dislocate the drug. And this is, from TB. The previous one was from HIV. Okay, so also in TB example, we see that there is quite a bit of change in the BC when you compare the wild type to mutations. And what you are seeing here, I just want to give one example. This is where the mutation is, this is where the active site is, and blue ones are the communication hubs with the mutation. And there is a path going, we believe that there is a path going to active site. And currently we are analyzing this communication hubs and checking how the interactions are changing in the active site area. Okay, my last example to mutation analysis is from I'm unable to say the name of these drugs. Pyrimethamine. Pyrimethamine drug resistance, and this drug is used in malaria, and this work was done by Arnold and Roland, and recently published in Molecules. This drug, sulfadoxine pyrimethamine, is an important malarial drug. It's used as preventative, but there is it binds to it targets the DHFR, but again there is resistance mutations, and these mutations can be one mutation, double mutation, triple or quadruple mutations. So we again model the protein with mutation and the drug and looked at globally. We again saw that root mean square deviation is differing in wild type when there is between mutants and wild type, and also in the presence and absence of the medication. So this is basically the histogram presentation of the root mean square, and it shows the conformational differences. The wild type is gray, and the green is wild type with the drug, and the other pink and blue shows the mutant protein. As you can see, the structure in this case is the overall global structure. In some of the mutations are highly different than the mutant one. But what is also interesting, what you are seeing here is the kernel density estimation graph for the behavior of the ligand. Ligand in the drug in the wild type has the model behavior. It flips. The one part of the drug flips from one side to other, And that behavior changes, the behavior of ligands changes in the mutant proteins. And when you come to actually the quadruple mutation protein, you see the largest conformational heterogeneity. And that is somehow expected because this is basically, these mutations are in the active site area. So there is nothing really challenging here. There is the other thing that we looked at in this drug was the binding affinity and the mutations. What we observed, that mutations weakens the binding affinity of the drug to the protein. Well, this is this value is for the wild type. As you can see that in the quadruple mutant protein, the binding affinity is much less. Again, the BC analysis, I'm just gonna show this figure quickly, and red and blue shows the, depending on the mutation. As you can see that the interaction, the communication pattern changes in the presence of the mutations. And communication changes also in the absence and presence of the drug. So drug has also effect in the protein behavior. Okay, so I have very short time left. Very quickly, I would like to talk about allosteric sites, alternative drug targeting sites. Allosteric in general definition is a functional change at one side of a protein caused by a change at a distant site. And allosteric systems were perhaps discovered over 50 years ago, but the concept of using allosteric sites as drug targets is still not common, despite the fact that allosteric drugs have lots of benefits over the active site drugs. They don't bind the active sites, and designing the active site drugs are quite difficult sometimes because the active sites are conserved, especially if you are working with parasitic diseases and if you have homolog proteins, host proteins, it gets really challenging. Hence, active allosteric drugs are much much easier to design in a way. Hence, and they are also more specific. It means that they have less toxicity. They would have less toxicity effect in the body. So again, when we start looking at this problem, we realize that there is not one well-established method or protocol. We proposed our own protocol, and which includes the identification of allosteric sites. Here we do a number of different things. We looked at, we use the existing tools for the cavities. We look at literature, of course. We use something called perturbation response scanning. This has been known technique, but the first time we used for the allosteric site identification. And of course, do the blind docking to see if the ligands are binding somewhere else than the active site. And then we confirm our results by doing MD and dynamic residue network analysis. When we identify the allosteric sites, we then go back and start doing virtual screening, again molecular dynamics, and then the conformational change analysis coupled with the network analysis. Okay, so here I'm just gonna summarize some of the publications that we had in this topic more than explaining the projects, because I have only 5 minutes left. These are actually the key articles on this, perhaps these two, where we established a methodology by combining the molecular dynamics coupled with perturbation response scanning and dynamic residue network analysis. So these three together can identify the hotspot residues. As a follow-up, and we used heat shock proteins as case studies. After identifying hotspot regions, potential allosteric regions, which would lead to conformational changes upon binding of ligand, we did a further study and looked at a human HSP90 for the cancer as a cancer drug target. Here we had two potential allosteric sites. We did a screening and we identified three natural compounds from our natural compounds library, SancDB, and as a control we used Novabiocin. What we identified here, and this is the PCA calculations, just the projection of the behavior of these proteins in the absence and presence of the compounds. Novabiocin is non-inhibitor, and we saw that these two compounds would give the similar behavior, and hence they are potential inhibitors. And we identified one compound would lead this protein to close, go from open conformation to closed conformation, and that's basically how the protein gets activated. Hence, we think that this ligand is activator. The other study was done by Arnold to look at, again, this is cancer study, and we identified one natural compound. This compound is known that it works, inhibits the cancer cells, but we were the first time showing that this compound actually binds to heat shock proteins. And if you read the article, you will see that basically it locks the functional cycle of these dynamic proteins. Okay, so one important point to keep in mind, these studies, these protocols are not straightforward and it takes months of work. So we were trying to identify some alternative approaches. In that respect, we collaborated with Gennadi's group in USA, and we looked at the possibility of using coarse-grained calculations instead of all atom dynamic residue networks, all atom MD calculations. And in this study, we showed that actually they are in agreement, so we could use the coarse-grained calculations to identify the modulators. And finally, I would like to wrap up my talk with referring to this review article that we published this year. Basically, this article brings all the existing computational approaches and tools to identify allosteric sites and allosteric modulators, and we also have a section where we explain our protocol, how we do the things. With that, I would like to end my talk and of course acknowledge all the funders and collaborators and CHPC. Of course, without CHPC, we couldn't do so many MD simulations. Okay, thank you very much.
Okay, thank you so much, Professor Ozlem. You kept to time, actually. You still have that we still have a lot of time for questions and engagement from the audience. So anyone?
Okay, I'm ready
for questions. Okay, for question, you can either type it in the chat box Below on the left, that's my left hand, or you can raise your hand. There's an icon on top with a human-looking thing that's raising its hand, and you can press that, and then we'll be able to see if you have a question. Or comment, question or comment. And just by the way, this was being recorded. Oh, Victor has a question. Victor, go ahead.
Hi, very nice talk, Ozlem. I was just curious. I mean, there's always when you do these kinds of studies where you use in silico simulations of protein behavior, wild type versus mutant and so forth, whether what you're simulating actually reflects reality. So the question is, do you have any experimental data? I know that your lab doesn't do wet lab, but maybe in collaboration with other scientists that show that the things that you simulated are actually happening in quote-unquote reality when you look at the behavior of these proteins, protein drug complexes, and so forth.
No, we do have some collaborations. I'm wishing to have more, but currently, actually, it's an ongoing collaboration with some groups in Sabancı University, and we are looking at rare diseases. So their experimental data and our simulations are in agreement. So that's what I can say. And we are looking at different conditions, MD simulations in different cell environments, and they are doing exactly the same thing in the cell level, and we are getting the more or less the same results. So, yeah, and in docking studies, we have some published articles actually. We do large-scale screening, and we identified, there is one article, for example, in PTR1, trypanosoma, and we identified like 10 compounds, and we took them out of 5,000 compounds, and we asked collaborators to check in cell culture, and we got 4 or 5 of them showing activity. So actually it correlates.
Okay, thank you.
Okay. And perhaps I should add one more thing. When we do these simulations, we try to repeat it. We don't do one time. We repeat it, and we do long simulations. If in Olivier's case, for instance, we did very short runs, but then we had thousands of cases, and they calculated, supported statistically. So one way or other, we have the controls. If it's not wet lab, either with combining different approaches or by doing statistical tests, we check if it makes sense.
Okay, thank you so much for that, Ozlem, and Victor, thank you for your question. We still have time for more questions or comments. Just repeating the fact that we're recording this webinar and it will be up on our YouTube channel. We have a channel, a channel where we have a playlist, particularly for our webinars. So if you missed any part of this or you need it to watch it at a later stage, you will be able to do that. Any more questions, please raise your hand. Please do type them in the chat box, giving you some time. I'm going to keep quiet for now while
No, everything was crystal clear.
So, Ozlem, if you don't mind, I think I'll share the slides, because obviously you did agree that we record this and we'll put it up, but are you comfortable with me sharing the slides with the attendees? Because some of them have asked if I can share them. For instance, David in the chat box did ask if it's okay for me to share the slides. Are you comfortable with that? Okay.
Yes, yeah. I mean, yeah, sure. Also, everything is published except TV work, and yeah, I mean, that's
I'll
fine.
share my email with David, and then he'll give me his email, and then I'll forward the slides. Okay, thank you so much. I'll give you my email. I see he's typing in the chat box. So I'm just gonna. So obviously, if there are no questions for now, you can email Ozlem if you want to have a discussion on the side. If you don't have an immediate question right now, so I will not wait any longer. I'll just give it 2 seconds. Someone is typing. There might be a question. Let's see. Ozlem, they want your email address. Sorry.
Yes.
The root one or the Gmail? I can type in the Gmail. Ozlem. Oh, you.
I give both. I typed both of them.
Email with you guys. So if you want to have a chat, you want to engage on her work, on her research, possibly you want to collaborate, or you have any further questions or comments, you can email her in your own time. Okay. So for Ozlem,
Okay.
thank you so much for this interesting and very informative webinar. I think it's been, for those that don't know, we've had a bit of back and forth because Ozlem is very busy and it's hard to pin her down and get her to actually say because we're November and something came out. But I'm glad that you finally could give a presentation.
Yeah, me too. Thank you. Thanks, Pablo. And thanks to all of you for
Thank you so
listening.
much. Congrats again for being a professor. Well done. Sure.
Oh, thank you. Okay, thanks. Bye. Some people are typing. Okay, so they're just saying bye. Okay, thanks. Thanks,
you.
everybody.
We'll see you next month. The last Wednesday of the month, we'll have a different webinar. And thank you so much for joining us, everybody. Enjoy your day.
Okay, bye.